Hello everyone, welcome to this Intelligent Infusions Masterclass. My name is Andreas Kiesling, I will be your host today. I'm part of the service team here at Cube Biotech, and I have a PhD in biochemistry at the University of Leeds. So it's my pleasure today to have someone from the lab that I was doing my PhD in. So Alex Snow is part of Stephen Muench's lab and will be giving a talk today about receptor tyrosine kinase and will explain how he solved the challenge of purifying the protein in a very complex manner. So today we also have Steve Muench himself, who will give an introduction of Alex Snow. And it's a pleasure always to have someone in the field of copolymer research at CryoEM. So that's why it's so great to have this connection still going on. And it's a pleasure that Steve, you can give the introduction and then later on stay for the Q&A so we can have a bit of a more lively discussion going on. So to you, to Leeds.
That's great. Thanks, Andreas. Nice to see you again as an ex-Leeds alumni from the group on level 6. So for those that don't know, a brief introduction to the group. So as Andreas has said, we're very much into structural biology, primarily around electron microscopy, as a lot of groups are these days. And we do a lot of method development around things like sample preparation, time resolved EM. But of course, structural biology doesn't work very well unless you have any proteins to go in the microscope. And so a large part of what we do as well is also on protein production and method development around there, new ways to sort of express proteins and stabilize proteins, especially membrane proteins. Hence, we've used SMALPs for a long time now in the lab and other copolymers. Some of the proteins we work on are easy. Some of the proteins we work on are really hard. The protein Alex is going to talk about, that definitely falls in the hard category. This is something that a very talented PhD student spent 4 years working on before Alex took over to do a postdoc. And it took him a few years to really crack this problem. It really has been a challenge. So it's great that he's been able to talk to people about how he's able to sort of crack this challenge and use the sort of cell-free system. I don't want to give too much away into the talk, but a very quick introduction to Alex as well. I've said he's a very talented postdoc in the group. He actually did his undergraduate at Southampton University before moving on to do an MRes at Imperial College, and then did his PhD not far away from Leeds over in York. So he's finally moved to Yorkshire with Professor Gideon Davis, where he's learned a lot about sort of biochemistry and structural biology, and he's brought those skills really to Leeds. And like I say, it's my great sort of pleasure really to introduce Alex's work. It's great that he can talk about it because, you know, he's the one doing all the work in the lab. I just sort of do the paperwork and the admin really as PI these days. So over to you, Andreas. Alex, have the screen.
All right, thanks for that.
All right, hi everybody. Thank you so much for that lovely introduction, Steve and Andreas. It's such a pleasure to be working on such a cool problem and in such a cool environment. So without further ado, I will tell you all about what I do. And I put it together into a title that I'm calling Cell-Free Protein Expression, Copolymers, and CryoEM. 3 reasons why that structure may be closer than you think. I am an eternal optimist, so that structure is always closer than I think. So off we go. First of all, we'll dive straight in and I'll tell you a little bit about what I work on. So I work on receptor tyrosine kinases, RTKs, which are a ubiquitous eukaryotic signaling protein class. And they have this generally conserved activation sequence wherein an extracellular ligand will bind to their extracellular domain. That tends to induce lateral dimerization of the receptor, so it forms a homodimer typically. And then that brings a pair of kinase domains close enough to one another that they can undergo trans-autophosphorylation, wherein they'll decorate one another with phosphotyrosine modifications, which can then be accessed and bound by downstream signaling adapters and proteins to enact a very wide range of signaling processes. Chances are, if there's a eukaryotic signaling pathway that you can think of or can work on, chances are there's an RTK in there somewhere. I cannot work on them all as much as I may like to, so I am focused, and I'm focused on the fibroblast growth factor receptor 3, FGFR3. Which is an RTK that is involved in PI3K, MAPK, and STAT signaling pathways, among others. It has a very wide range of uses within the cell and within the greater organism that include everything from development to homeostasis, tissue repair, growth, just about any kind of regenerative process they tend to be involved in. And you can see just how wide a type of signaling that they can have, because FGFR3 alone binds one of 18 different FGF protein ligands, as well as a heparin sulfate cofactor in the case of paracrine signaling. So it's a very wide range of signals that it can receive, and it somehow transmits all these signals across into the cell, and they have disparate effects. Naturally, this protein is subject to mutation, and it has mutations that have been mapped across the entire protein, so in all of its domains, the extracellular, the transmembrane, and the kinase domains. And these cause a range of diseases, notably cancers, because any protein that is involved in, cell replication, if it goes wrong and becomes constitutively active, that's obviously going to start causing you problems with unrestricted growth. So naturally, it makes a very tempting target to look at in any kind of detail. And since I'm a structural biologist, my thought is, well, where's the structures? What can we find on the PDB? So structures do exist, but they exist only for the separate domains. So I've put together a range of structures that you can see here. Extracellular domains, which you can get by X-ray diffraction, the transmembrane domain that has some NMR structures, and the kinase domain that, again, is mostly either by NMR or X-ray crystallography. And what you might notice is that all of these individual domains are separate from one another. People tend to solve the individual domains as truncation variants. And that makes sense if you're trying to get, any kind of structure, because if you're trying to crystallize a membrane protein, that's obviously quite the challenge. So why do that if you can just study the extracellular domain or whatever else have you? The problem with that is that you then don't have an idea about how a change in one domain affects the other domains. We don't get an integrative view of the structure, which would be immensely valuable because it would help tell us a lot about exactly why the receptor goes wrong. In a lot of the cases it does. Right now we get a very limited view comparatively. So if it's so important, which I'm certainly talking it up, why doesn't that actually exist? And it doesn't exist because it's kind of like Steve said, you can do all the structural biology you want if you've got the protein. And FGFR3 is very hard to make. I spent the first 2 years of my postdoc, and the PhD student before me spent a good deal longer as well, trying to make this protein. And we didn't have very much success for a very long time. So I first off tried in SF9, just in sex cell media, doing a baculoviral expression system, fairly standard stuff. And I attached a GFP tag to the protein, the idea being I can visualize where it's being made and how it's being made and how my virus is doing, essentially. And the protein was produced, yay, but it's not in the right locations. It ends up in early endosomes, and it should be in the plasma membrane, but it seems to end up everywhere but the plasma membrane, which is obviously not ideal. It doesn't give us a large amount of hope for the structure we get being something that's actually physiologically relevant if it's not being in the correct places. So I then tried HEC293. I tried a BACMAM system, and that did again produce protein after a great deal of optimization, but it was never functional. What I found was that the expression of the protein usually killed the cells when I was making them, as well as the virus inevitably killing the cells too. So I had this very narrow window where I could actually make my protein and harvest it and have anywhere near anything that you could think would be physiological relevance. It's not in broken down pieces of cells, it's in actual cells. That's the kind of grain we're talking here, let alone anything, where you could test it physiologically. So even when I got it out, it was just never functional and never good. So overall, my first 2 years of my postdoc were exactly what you like out of a first postdoc, which is an ineffective and time-consuming and ultimately producing poor quality protein. Wonderful, just what we want. So my first thought was, okay, if the usual things aren't working, why don't we try something that's a little bit more unusual? And that brings me into my first point, which is for protein expression, sometimes you got to think outside the box a little bit. So we went to cell-free protein expression, CFPE, which has a bit of a, shall we say, a bit of a reputation in the molecular biology world as this thing that you might be able to make some toxic proteins with, but it's quite challenging and it's got all of these problems. And, how are you going to get an RTK out of this? And I think the first step in maybe dispelling some of that is actually telling you a little bit about what a CFPE is, which is often case an optimized cell lysate. So what you do is you take living cells, be they E. coli, plant cells, mammalian cells, whatever your starting stock happens to be, and you create a cell lysate. And there are varying levels of technologies that have gone into this. You can see papers from ages ago where people just made them using their own ultracentrifuges, and now you get much more advanced companies that are making these things to a much, much higher standard. And the principle is you're trying to choose what you keep from cells, and you're trying to remove the things you don't want from cells. So you want to keep translation and transcription machinery, otherwise it's not a protein expression system. You typically actually engineer these things so that they are compatible with T7 systems. That's fairly common, so that you can put a adapted T7-based vector in and then get your protein to be expressed that way. And then you often keep soluble proteins. You tend to lose things like membrane-bound organelles, nuclear mitochondria where applicable, unless you're deliberately adding in things again, like membranes. Something that's crucial for my work was that you lose things like transmembrane signaling pathways. So you lose any interfering signals where, if you're trying to make, say, an oncogenic version of an RTK, you get signals that are telling the protein, telling the cell even, degrade this, get rid of this, or kill yourself. There's something that's making us toxic, so we're going to put in all of our, cancer defense mechanisms and kill the cell instead. You get rid of all of that, so it's just making the cell, you're making the protein even. You get rid of things like localization, again, unless you deliberately engineer them back in, typically. And you also get rid of a lot of regulatory mechanisms, like I just mentioned, and things like genomic DNA. So you end up, hopefully, with a sort of optimized lysate soup that can act as a protein factory, as opposed to a living thing that you've co-opted into making your protein. These have been used, as I said, primarily for things like toxic soluble proteins for a long time, but they actually have a lot of applications for structural biology. And this comes about, I think, for the biggest reason being that you can actually optimize what amino acids you put in. Instead of having all these regulatory pathways or your carbon catabolite repression, things like that, that are, down-regulating certain pathways if you add a certain amino acid precursor in, you can just add in the amino acids. If you want to add a non-standard amino acid, say radio-labeled amino acids for NMR, then you can just put them in. And if you have the right kind of lysate and you have the right kind of method for that, then you can incorporate those fairly efficiently. And what you're seeing here is some protein NMR of a membrane protein that was produced by a cell-free system. So they've got a huge advantage there, and I'm glad to see that the world's kind of picking up on that and going, actually, this is quite useful, this is quite interesting. Why don't we try this rather than, say, radio labeling a bacterial culture, or worse yet, trying to do, mammalian TC with a radio labeled media, which is unbelievably expensive if you've ever considered it. So I'm glad to see that's making some progress. And if you're not an NMR person, say you're an X-ray crystallographer, the same thing is true too. So say you need to do some experimental phasing, you could use something like a selenomethionine label, rather than having to do, say, a magic 6 style, pathway inhibition method where you're trying to, downregulate the methionine biosynthesis pathways. You can just add your selenomethionine straight in, and it should, if you have the right system, just work. Again, this would be very challenging in, say, mammalian cell culture. So for any protein that requires that, this can be quite game-changing. And again, I'm glad to see that people are picking up on that. I've been alluding to mammalian proteins and mammalian tissue culture, and this is because, of course, eukaryotic proteins feature things like complex PTMs that E. coli can't replicate. That's why systems do exist for systems that aren't E. coli. So you can get wheat germ, yeast, human, plant, and you can have different successes depending on which type of lysate you might use. But here's the thing, even if you have a system that can't necessarily be recreated, or maybe you can't for an overexpression, like say an acetylation or a myristylation, then you could add the relevant transferases in. So the paper that I've got here actually describes them adding in, I think it's an acetyl transferase as well as a myristyl transferase to, which protein was this? This was alpha-synuclein, I believe. And they get some mass spec that shows, oh, we actually got it to incorporate, and we managed this through a cell-free system. And perhaps they can do that in a much faster turnaround than you would by, say, having to do it through bacterial or, mammalian cell culture. Overall, it massively speeds things up and just makes for a much, much better, I believe, system for a lot of these kinds of applications. So definitely don't sleep on it. Like if you have a protein that's really hard to make, try something like this. So let's say you've got your protein and you've tried your cell-free system and oh my goodness, it works. This is where I was about 4 months ago. Oh my goodness, I can get my protein. That's incredible. Now I've got to purify it. Because it's a membrane protein, I've got to make it soluble before I do that. So how do you manage that? Well, I'm sure anybody who's at a Cube Biotech meeting is familiar with the concept of, say, SMA or other copolymers. And these offer a detergent-free option where you can excise a protein from the membrane but keep the surrounding lipids. Oftentimes, lipids can be quite important for how your protein is actually functioning. You'll find that you have lipid binding sites within proteins. You might not be able to reconstitute activity without those lipids present, or it might modify the activity. And of course, that's something you want to study. So you need those membranes to be there. Now, sure, you could say do a detergent isolation and then make, an MSP nanodisc or something, but then you're having to guess which lipids you're putting in, because you're having to put in, say, a mix based on human plasma membranes, which might not be the same as whatever specific lipid environment your protein actually translocates into. You may find it has a local, increase in one particular one, or maybe more cholesterol or whatever else have you that you can't necessarily anticipate if you're doing it through a method where you're having to then put in the lipids yourself. So having a system where you can just cut it right out is very, very useful. That is, of course, easier said than done, which is why, luckily for us, a range of these kinds of copolymers exist, everything from SMAs, dipmers, amphipols, all of these kinds of things. And not all of them work for every application. I'm going to go into this in a little bit and tell you a little bit about some challenges that we face in the CryoEM world for this. But my immediate take home here is it's worth experimenting. So, you may find that you have a copolymer that works very well for mass spec, but does not work for EM. Just because you get one thing that works, don't necessarily just stick with that one thing. Do screen, do think, oh, it works in this one and this one and this one. Maybe the efficiencies are slightly different, but if one of these will work a lot better for your application or for different applications, certainly don't sleep on that. Again, definitely optimize for that, because it's absolutely worth sort of spreading your attention as far as you can there, I think. So if we're going into the CryoEM world, CryoEM has some specific challenges, and they arise a large amount from the way that you actually produce a CryoEM sample. So for anybody familiar, you're essentially adding a very small amount of your protein, usually about 3 microliters, onto a grid, and then blotting it off and immediately vitrifying it, or vitrifying it as quickly as you can. A vitrobot will do it in a couple seconds. Some of the more interesting technologies that we actually work on in our group here can do it on the millisecond time scale, which is very, very cool. And this is really important because you end up with this issue at the air-water interface. It's a thin film effect that you get on the grids themselves. Now, Steve's the expert, and he can explain this obviously a lot better than I ever could. But some big problems that you have in the context of copolymers is that you get these effects at the air-water interface where you have a lowered pH and an increased divalent ion concentration. Now, these can often make smelts start to degrade. So, for example, SMA can be quite pH sensitive and quite divalent cation sensitive. And if your protein is starting to fall apart when you put it on a cryo-EM grid, obviously what you then study is not going to look amazing. So again, experiment. There are different SMA copolymers out there, different copolymers in general, that have different resistances to these things. You may find that you need a specific one. You may find that you need to immediately solubilize into something that is more aggressive than the thing you're going to end up in, and then exchange into that final amphipol, copolymer, whatever have you. That's a reasonably common workflow. And it is because you have different amphipols for different use cases. So we could talk about detergent as well here. DDM, you could say, lacks the issue of, lowered pH and increased divalent cations. It's not as sensitive, right? But you have a higher background because you'll have empty micelles all over the place, and you have to have a small amount of DDM in your buffer in the first place to prevent your your micelles from destabilizing, essentially. Increases your backgrounds on EM. Again, also, your particles will lack those important lipid contacts, so what you might get out as a structure may not be as relevant as what you could get if you were able to maintain that lipid environment. So you can see some very obvious differences that you get in your particle distributions just from these micrographs here that show in different materials, amphipols and DDMs, and before and after freeze thawing. I would never recommend freeze thawing a membrane protein before you put it on cryo-EM, even if your protein is stable enough to, in theory, survive it for other uses. Really, I would purify it and use it in the same day, which makes for very long days, but it's worth it, right? If you see some nice 2Ds and things like that. So definitely worth doing that. So finally, my third point is, say you've got all of these things down. You are in a situation where you've made your protein, you've solubilized into something, you've purified it. That doesn't necessarily mean that you're going to get a cryo-EM structure out of it, though. So you might need to think a little bit about what you're studying and the technique that you're looking at it by, and think, how can I make these things match each other just a little bit better? And if we think about that in the context of FGFR3, FGFR3 is essentially a series of small individual domains. So it's 3 Ig domains for the extracellular domain, followed by a transmembrane single pass alpha helix, followed by a kinase domain, and that will dimerize to form 2 copies of that. But then there are flexible linkers between all these domains, and you can stabilize some of those in the extracellular domain by adding in your ligand, but you've still got something of a pool noodle, as I often end up calling it, where it's just kind of flopping around. There's an awful lot of conformational heterogeneity there, and it's certainly not what you want for CryoEM. You want something that is as stable as it can possibly be, is in one conformation that you're going to get some lovely 2D class averages out of, and that they will all be representing one particular conformation of your protein. So the question is begged here, can you study something like FGFR3 that is this pool noodle if you have these series of domains with flexible linkers? And I think yes, because again, I'm an absolute optimist. But I think it might be better if you tune what you're looking for and look at something specific. And that's where a particular mutation comes in that I'm interested in, which is FGFR3 TAC3, which is a chromosomal rearrangement which causes fusion with transforming acid core containing protein 3, TAC3. And there is a variant of this called RT112, which is found in bladder cancer. Now, this variant undergoes ligand-dependent dimerization, but it seems to undergo ligand-independent kinase activity. And the reason I decided to study it is because, one, it has this fairly well characterized, this is directly from primary cell lines that this activity has been measured. It has fairly well characterized activity. So I should be able to tell whether the stuff I make by my cell-free system or not is actually applicable. Is it physiologically relevant? Is it doing what we expect? But additionally, the TAC3 is a core domain. It's quite long, quite rigid. And my hope was perhaps it will anchor the intracellular domain, the kinase, somewhat, so that instead of having it flop around on these long flexible linkers out of the membrane, it might be locked into one specific state. There are further proteins I'm working on as well that I'm going to try and introduce into the system that are much future plans and a bit hush-hush right now that might be able to stabilize this further. But I thought, with our starting protein, let's try and see whether we can get this to be as rigid as we can possibly be. So where did I start with this? I started by doing some cell-free of RT112, and I used an ALICE system, which is by a company called Lenio Bio. They pick a plant cell lysate, and I found it's been working for me quite well. Worth experimenting, as always, but this seems to work pretty well for me. And we tried this with about 24 nanogram per microliter concentration of the DNA that we're adding. The DNA is in a T7 regulated vector. And this DNA, I'd always recommend optimizing this kind of thing if you're going to do something like cell-free. It's like anything else, make a microplate style reaction and try and find out what's going to work best for you, that sort of thing. Anybody who's familiar with tissue culture might be looking at a 48-hour expression cycle and think, oh my God, how did you manage that? That's amazing. And it is, isn't it just? Rather than months of making a virus and precipitating it and then titering it and doing plaque assays and things like that. Instead, you add it 48 hours later, you get an idea as to how well things are working. I found 80% of my protein was being made in 24 hours and then that further 20% in that 48-hour time point or so. I've not measured pre-purification yield, but I tried a fairly rough and ready using some in-house stocks of smelt that we've got. I tried sort of in rough and ready, will this thing solubilize? And it did. And I found that we're getting about 200 micrograms per 1 mil of cell-free lysate, which is quite a good yield for something as complex as this. So I was quite happy with that. We then moved on to some solubilization. And I have, in fact, tried with DDM, just a standard detergent, right? And I found that it will solubilize in DDM. That was just sort of a positive control. If it can solubilize, will it solubilize here? And to no one's surprise, it does. But it didn't purify very well. I found that I was getting a large amount of variety in the number of copies I'd get per detergent in my cell. I was getting a lot of conformational heterogeneity. Essentially, it was a bit of a mess. So I also tried it with some SMA, and I found that as a solubilization step, that worked pretty well. So that, again, purified quite well. It's been forming quite a, stable single fraction. But SMA 2000 is quite pH sensitive and it's quite divalent cation sensitive. So when I go onto some CryoEM, I've been getting a lot of stuff that falls apart. It's not been ideal, really. And that was causing me some issues, obviously, for CryoEM. So I decided, let's try some other methods here. So that's where I get into this idea of maybe exchanging into other copolymers or amphipoles, things like that, things that might be finer grain, things that might reduce that, falling apart somewhat and maybe be a bit more rigid, perhaps even, be able to distinguish a single conformation a little bit better, increase that, homogeneity, which is, of course, what we're all about in CryoEM. So the future is bright there, essentially. So that is the same as that. And then we finally come to, does it actually work, what I've produced and purified? So we chose a ligand, FGF1, and we thought, okay, we're expecting this to dimerize our protein. So can it bind and stabilize the receptor? And does that then induce this dimer conformation? So I tried a couple of solution molecular weight technologies here, mass photometry and native page, and these both show the exact same story. So they both show this ligand-free population that is forming approximately a monomeric molecular weight. And then when you add in your ligand, your FGF1, you find that we're getting still a sort of monomeric population there that's maybe a slightly higher molecular weight is bound that ligand, but we're getting a much increased dimeric population suddenly start to form, i.e. FGF1 is binding to the receptor and then causing dimerization. So far, so good, right? It's working as we expect. Well, the problem has always been the kinase domain with these kinds of proteins. So this was the bane of my life when I was doing, the mammalian stuff, because it would never ever be active, and the kinase domain would never work. And it should be constitutively active. It should be working regardless of ligand binding, regardless of almost anything. So does the cell-free method actually produce active protein? And this was a slightly challenging thing to look at, because unlike, say, a substrate kinase, where you just add in your substrate and ATP and then look for ATP turnover, it's autophosphorylating. Its substrate is itself. So how do you measure that? Well, what you do is you get rid of all the phosphates that might be on the protein by adding in some lambda phosphatase. So you strip them all off of the kinase domain, and hopefully it's just in a fresh state where when you re-add in ATP, either you will get no further phosphorylation occurring, in which case when you couple it to an ATPase fluorescent assay, like an ADP glow, you produce no fluorescent signal. Or if it is active and has been phosphorylated, you'll get ATP turnover, and that ATP turnover will be detectable with fluorescence. So which do we see? Well, very happily, we see activity. So what we see here is a very significant increase in ATP turnover with sample that has been treated with lambda phosphatase versus sample that has been untreated. So the idea here is the kinase activity was dependent on us stripping off those phosphates before. This is impurified protein. So the idea is our protein is undergoing active ATP turnover, phosphorylation. It is an active kinase, and this is independent of any ligands added, thus constitutively active kinase. I cheered when I found this data. I was very happy about it. So if I'm to conclude these points that I've been making, I'd say if you want success stories, and I'm hoping this is going to continue to be a success story, I'm certainly very happy about it so far. If you remember a protein doesn't seem to be expressible in other systems, try cell-free. It may well end up helping. If you find that you have a specific structural biology problem that requires, say, a labeled, non-standard amino acid, it's sort of ideally suited for that. So definitely worth looking at, especially if, say, you need to do experimental phasing on your problem and it's a mammalian protein. Give it a try. It's a lot easier than making up all of these viruses and spending all that time doing that kind of thing. Once you have the protein, you need to solubilize it, and that should be tuned for your use case. So there are a lot of copolymers, a lot of detergents available. So definitely screen, and definitely don't just find the one thing that it solubilizes best into. Do think about what application you've got and where your copolymer or amphipol or even detergent is going to be best suited for that. And then if your target is naturally hard to study by whatever technique you're looking at, think about whether or not you can modify your question somewhat. Can you look at one specific mutant of your protein? Can you look at it in complex with something else? What can you do perhaps to make your target slightly more amenable? What's going to get you that data? Because after all, you can be bashing away at a problem for years and years and years, but is that going to help as much as, say, getting a slightly different question answered that you can in fact publish about and then maybe get that grant and then maybe keep at that original problem for longer? Perhaps thinking tactically there. So finally, I'd like to acknowledge, of course, Steve. I'd like to acknowledge a very talented master's student who's been working with me on this project, Tharashi. Thank you very much to her for all the hard work there. And then everybody else in the Breeze Lab as well, who's also working with me, the EM facility here in Leeds, the Catan Lab in UCL, the NMR facility here in Leeds, and finally my funding body, the Medical Research Council. And I've included a QR code here, but actually you can just press the button up top if you'd like to read our preprint on this. So thank you very much. And do I have any questions?
Thanks a lot for that really interesting and entertaining talk. I think that's very rare in science that you could get good entertaining talks and have a good behind it. Very refreshing. Yeah, maybe in the meantime, people can pose questions, but I think also from a personal history, if I remember the late nights in the lab coming in and seeing then a very tired PhD student seeing again that a protein is precipitated and then trying the methods all over again. I think what was interesting quite here is that it needed the courage to try new technologies. So one, the cell-free approach for the expression, and also then the copolymers to get that project actually done. So it's not only a question of, okay, you can try very hard with the more established method, but it's also you needed to have these new technologies to get that project to the finish line. So that's my takeaway from this.
Absolutely. I now believe that if I had 6 years to work on this through mammalian cell culture, maybe I could have gotten to the point where we are now. But it's the fact that I had that fast turnaround, and as you say, that sort of 1-2 punch of being able to go, here's this very new cell-free technology, and here's this very new copolymer technology. Combine both, use the strengths of both, and we end up somewhere that perhaps we couldn't have ended up with any other way.
Right. Yeah. Yeah, I think what also was interesting, you were mentioning that a bit with the flexible linkers and the domains, because traditionally, okay, you can truncate your protein and you can look at the individual domains, but really to stick to the full length and really wanting to know the interaction between the individual domains is also quite an important part of the project, I would say.
It's vital. So it's a problem with RTKs in general. And I think a lot of these quite flexible signaling proteins is that no one has studied them structurally in this way because it's so difficult to, and yet it's so critical. I mean, I touched on the fact that FGFR3 has so many different pathways associated with it. And there are some theories about how that signal is being transduced across the membrane, but there's nothing concrete because we don't have that. Here's a change in extracellular that leads to change in transmembrane that leads to change in kinase. So having a basis by which we can potentially study that, I think, is a very potent thing.
Right. So there's a question coming in. I can highlight that question. So someone is asking about why you choose Alice as a cell-free expression system, maybe to give you a bit of a background about this.
So we had a contact in the company that was able to help advise us on use of this technology. So it made something that at the time to me certainly was very obscure. You know, cell-free wasn't something I was familiar with at the time at all. It made that a much more understandable thing because I was able to say, you know, hey, isn't this a technology that's only used for, you know, soluble toxic proteins? And they were like, no, actually we have methods of making membrane-bound proteins. And we have methods of doing this and doing that. So we were able to very much leverage that sort of contact and community, and from that get ourselves a kit, and then from there be able to test it out, essentially. I have not tested with other companies simply because I found this one's working, and it's working really well. I always recommend testing things around in any use case for everything, but I would say if you're looking for a first use case, they are good people to go to, and you know, certainly worth it.
I mean, if you spend 4 or 5 years on a project and suddenly something works, you'll stick to it. So
absolutely. And the second you can be done with a problem, you want to kind of be done with that problem, right? And you know, the second I'm finding, oh, I can express this protein and it is active, I'm like, well, I'm not going to shop around now. I'm going to keep for this unless I find that there's a use case that requires me to do something different. Yes. So if I find that there's a use case, then that would be different, but that would be different in everything.
Yeah. I mean, in the end, it's not about the technology, but how you get to the end product, which is your protein. And then you want to solve structure or use it for assays. That's what you're actually interested in, not to how you got there in the end.
Exactly. It kind of feels like reading a book from the end, from the back page, you know, oh, you got the structure, happily ever after, amazing. How did I get there now? What do I do? Which is, of course, the optimism of he will get the structure in the end or whatever else have you. But you know,
let me just see how I get these other questions in. Danke. So how have you tested this on other oligomeric membrane proteins?
Not yet, out of time, but it is on my to-do list.
Right. So the kinase does have interaction partners, but they are cytosolic interactions partners, if I understood it correctly.
Yes.
Yeah. Okay, that makes sense. Okay.
It's an issue that I've had with cell-based systems, particularly HEC-293, back when I was using that, is I'd actually pull a lot of those down when I was purifying.
Right.
You'd get various interactions that I suppose the oncogenic variants just make that strongly. So it was another reason why I was never able to really purify that protein very well, because it would just pull down all kinds of things, and I'd get such a high degree of heterogeneity in my sample. It was almost useless to study.
Yeah, I think also maybe it's here important to highlight the use of copolymers, because if you think about detergents, if you have a weak interaction between your soluble partner and your membrane protein, that you always have to have some background level of detergent present, and that will then also impart on that interaction. So having a copolymer there, which doesn't need anything in the background, can then also help with that interaction.
Absolutely, and that's certainly what I saw when I started moving to copolymers, and it's something I continue to see as I continue to experiment with them, is you generally get a much cleaner result for that reason.
Great. So are there membranes in the cell-free expression system? So in Alice. So if not, what happens to the hydrophobic transmembrane domains?
So there are indeed. They are plant microsomal, and the vector that I use actually inserts them has machinery to insert them essentially into that membrane. You're then able to extract them as though it were a membrane protein from, say, similar to a direct cell solubilization, if anybody's tried that, you know, where you're adding either a detergent or a copolymer directly to your cells in order to solubilize it. I find that works.
Okay, yeah. Let me just, thanks. So if it's a plant-based in Alice, and then you extract it into SMA, you will be getting, or you're getting plant lipids with a human protein. Yeah. So I guess the behind question there is how that then impacts later on the structure solution and the assays.
There's the thing, we don't know how much that impacts on this protein. Say you have a protein where you absolutely know that it must have human lipids. Then perhaps using a human-based one might work better for you. I believe some of them you can actually add in your own lipid mixes, but you're once again making that assumption that you know what your protein is going to insert into, which may not be 100% accurate. So again, that would be a case where I would say, you know, tune what you're doing to your use case. You know, you may want to find something that will work better if you know that there's going to be critical human interactions. As it is, we've not done lipidomics on this yet to suggest whether or not that would be the case here.
So if I remember correctly, then membrane proteins also recruit the lipids that they need for their stability. So in a sense, it then takes the lipids that it requires to be stable, at least to the point that you can get it onto a grid and for your assays.
They do. I guess it's a case of, you know, does your system actually contain the lipids you need? To give you an example, I used to work on a plant cell lipid in my PhD, which it essentially does not exist in humans, but it's very common in plant thylakoid membranes. So say you were making a protein that was involved on that, you know, a photosynthetic protein, for instance, then you'd need to think about that. You know, you wouldn't be able to say just use a standard human one, because you might have those crucial interactions that you're missing because you're missing that specific lipid.
So I guess also, is there a need for like a orthologous method where you look at like a cell assay then to verify that what results you're getting with the extracted protein?
I would imagine that it's something you do as part of the course of characterizing your protein, whatever your protein happens to be, because what does activity look like for your protein? Are you doing ATP turnover as I am, or is it a, you know, say voltage gated ion channel where you might do some patch clamp, or if it was whole cells, or, you know, otherwise some other technique for looking at it in a more reconstituted manner? I'd say definitely study it. but exactly what that's going to be, I don't imagine would be standardized.
Right. Okay. Oh, okay. So just for time's sakes, if we go past the time, then all the remaining questions will be answered by us by email. So always feel free to ask, even if it seems that the time is running out.
Absolutely. I'll be very happy to answer any questions by email and have any discussions anybody would like to as well. Very, very open to that. This final question, Darren, was your protein codon optimized? Actually, human codon bias. And I kept that specifically because of the kinase domain. My reasoning was we were never able to capture activity in human cells, let alone, the insect cells I was really trying. And I think it's because that kinase domain was fragile enough that it really needed to be translated as close to native as it could be. And I was very happy to see that the cell-free system was able to manage that. But I do wonder if I'd codon optimized it, whether or not I'd have actually lost that activity just because it wouldn't have the same,
Yeah, if I remember correctly, then codon optimization is also a bit sometimes of a double-edged sword, because sometimes the folding of a protein is encoded in the codon usage, and if you then optimize it to another system, this information gets lost. But of course, you're going to another system, so it's always worth trying out different approaches and seeing if that actually makes a large difference in expression.
Absolutely. I would say if you're going to try something like this, honestly, my advice would be to start without codon optimization for that reason. You don't want to start changing so many things that you don't know why your protein isn't working. If you've not codon optimized, and you can say, what's the system? Or maybe I do need to, that kind of thing.
Right. Okay.
Yeah.
So any hands-on experience with which polymers seem to work better in CryoEM for resolution and sample behavior?
Amphipols, in my experience, tend to work quite well for that. Again, you're looking at things that are less pH sensitive, less divalent cation sensitive. So I would try, that's the experience I've had with other targets, is that an exchange into an amphipol can help. But again, these things are going to be so heavily tuned to which protein you're looking at specifically.
Right.
I could name an amphipol I've used, but just because it worked on my thing doesn't mean it's going to work on yours. So test, screen, essentially. Find anything that you think is pH or divalent cation resistant to a greater degree. And that might be a good place to start.
So just to clarify, because you said exchange into amphipols, I think you refer to the let's say older generation of amphipols, where you first needed to extract the membrane protein from the membrane and then put it into amphipols, because they're also the newer generation where you can directly extract it from the membrane. So just that because then people don't get mixed up about.
Fair enough. Yeah. The target I was working on did use older generation kind of stuff, so that was the method. But yeah, no, as you say, there are definitely ones these days that makes that a lot easier for, because making an exchange protocol work and optimizing that is a challenge in of itself. So anything that makes your life easier, that's good.
Okay, thanks. I think that was all the questions for today. And thanks again for this really great talk. If there's any remaining questions, please post them in the chat, and then we can later answer that for you. I wish you a good day wherever you are, and take care. Bye-bye.